Okay, so let's look at the phylogenetic classifications. And so in the phylogenetic classifications, this is a cladistic classification based on a common ancestry. And so typically, a phylogeny is an evolutionary history of a group or a species. So we can see that there's some common ancestors, but there's some branches in there. And so what we'll see there is when we have these little connections here, these are showing a common ancestor. With a new species, species A, B, C, and D are all separate species, but we have a common ancestor here, showing through this phylogeny. And so usually this is found because of evidence of living species that we have, the fossil record, and molecular data. Now remember, molecular data, we're talking about DNA. And when we have this, we can show the branches in a tree. Just like if you have a family tree made out, you can have a phylogeny from there too. Where your tree branches, like your parents will have a branch, you will have a branch coming off of there. You have common ancestors, parents, like you and your siblings, common ancestor would be your parents. Your parents would have your grandparents and your great-grandparents and so on and so forth. And so all of these branches equal this. And so this is one way of classifying by using a Clyde, which is a group of organisms that include an ancestor and all of its descendants. And so those are prime. So cladistic is a common method of classifying evolutionary trees based on a common ancestor. Species placed in order of which they descend from that common ancestor. And so you'll start to see things coming off, just like if you would talk about a phylogenetic tree, you're going to see that we have an armadillo here having its common ancestor, which is the species that is extinct now. And so the common ancestor will be like the trunk of your tree, and then everything else can then branch off from that. Genetics is a good example of how we can determine these cladistic evolutionary trees. And so basically it represents from this, like we can see our little armadillo here, we have branches such as the reptile. Clyde includes birds. And so we can see that the Linnean classification, mammals, reptiles, and birds are all in separate classes. However, we can use a cladistic evolutionary tree to see that there's one common ancestor in which they all kind of branch off of, and reptiles are the basis for mammals and birds, not just one. And so a Clyde, again, is a common group of species. And so we can see here, each species in the Clyde share a trait from an ancestor. So these are all different types of plants. We have our flowering plants, our cone-bearing, ferns, mosses, and then these are non-vasculars. And so these the non-vasculars were first, and we know that through the evolutionary trees that you did. Then we have our mosses, ferns, which are our first vascular land plants. Then we have our cone-bearing, and then we have our angiosperms, or our modern flowering plants. These are going to fall in the evolutionary scale of where they came from, and you guys should know that from the time scales that you did last week. And they come down here, we have a common ancestor. And this is where they all branch from, is this common ancestor. Now, we can see through DNA where they kind of all have some type of linkage, and they share a common ancestor. Just because they share a common ancestor, it does not mean that we know what that ancestor is. If it has not been discovered in the fossil record, we can only use molecular data to prove that they're linked genetically, but we don't have the common ancestor's actual name down for it. And so each species in this Clyde have a trait that has changed just a little bit, so that it has become genetically different from its previous ancestor, such as we have here non-vascular plants, and here we have a vascular plant. All of these have vascular systems, which means they all have a xylem and a phloem, but this plant does not. Okay, so we might not know what they is. So these derived characters or traits are shared in different degrees by the Clyde. And basically, with this, we can use a cleidogram to show more closely what the relationship is. And so here's our cleidogram, and so we can see where the derived species or derived characteristics have come from this. And so let's see what we have here. We have our 4-limbed species right here. And so this is showing that they have 4 different limbs. Okay, well, all of these up here have those 4 limbs. 1, 2, and 3 all have 4 limbs from that Clyde. Amniotic protective fluids. Well, that makes sense. These guys came from an egg, which had to be immersed in water, where all of these had either an embryo, which we have an embryonic fluid within the mother, or an egg which holds embryonic fluid and doesn't need to be in the water source because it has a hard shell. Skulls with openings behind the eyes, and then we have openings from the side of the skulls, skulls with openings to the front of the eye and the jaw, and then feathers and toothless / beaks. And so we can see where these derived characteristics have all come from. They've all changed. There are all these little slots here on the scale. So all these little slots and characteristics are these derived character traits from this classification system. And they lead to all these different developments of just these different species, which have some type of relationship. And so those cleidogram, which this is a cleidogram, the whole thing, is represented. We have these derived characteristics represented by these hash marks here. And so the cleidogram is actually showing all the species and then their derived characteristics as well. Nodes represent the most recent or common ancestors, and the Clydes can be identified by the separate branches or snips under each of the nodes. And so we can see our nodes here are all different colors. And so these are going to be your different nodes here represented by the colors and these lines coming off of the side, or these bigger branches. And so these are the nodes that we have here with the derived characteristics showing up underneath of them. And so this is just another way of breaking everything down so that we can see what we have. Now note, in our nodes here, we have our reptiles and our birds together, our snakes and our small reptiles, tortoises and turtles, and then we have 2 mammals and then amphibians here. And so those are the nodes, and that's a derived characteristic. Again, the colored groups are the nodes. The derived characteristics are going to be the hash marks between them. And then the Clydes are going to be represented by the names that are pointed out right there. Okay, so what is molecular evidence? Okay, molecular evidence is basically showing you what we have. And so this is going to be your DNA and your RNA. It's not just going to be your classification based on similarities, but it does confirm them. And so we do know that some things that look alike genetically are completely different. And so with this, we have a before and after. So this is with just our physical similarities. We had flatworms, roundworms, mollusks, segmented worms, and arthropods. So that was before. Once we had our molecular data, scientists realized, hey, guess what? Flatworms and mollusks are still here, but we're going to move segmented worms up here. And the roundworms and arthropods are actually more closely related. And so we did see some switching of some things once we have this. In fact, when we figured out this molecular data, we have a whole new phyla called Protista because we didn't know where else to put them. It's like the misfit group. Protista is actually developed because of molecular data, and we had to find somewhere to put that. And so DNA has given scientists pretty much the last word of saying, hey, it's related, or no, it's not related. The last thing I want to talk about really quickly, I'll go back to this, is you are going to do a lab based on classifications using a taxonomy guide. And so a taxonomy guide is a way of classifying animals based on kingdom, phylum, family, order, genus, and species, but you are going to answer a series of questions based yes or no. And so tomorrow you're going to go through a guide and it's going to explain this. But basically with a taxonomy guide, it's going to ask you, does it have feathers? Yes or no? Well, if it's yes, then you go on to another set of questions. If it's no, you go on to a different set. Basically, it's using the Linnean classification and the phylogenetic trees. And it's allowing us to ask a set of questions, yes or no questions, and determine what the species is at the end. This is also important because most of the time before this, we really use such broad or very specific questions, and we weren't able to figure out what species were. So once we came up with our modern taxonomy guide and these phylogenetic trees, we were able then to come up with a taxonomy guide where we could actually ask yes or no questions to determine what the species is. And so you'll be doing a lab based on that tomorrow. Okay, have a good day, guys.